Hello, Dr. Hudson. This is Megan Lahan, part of your online cell biology course, and this is my presentation on my assigned protein, DNA ligase. While I won't be able to read my paper word for word during this presentation, as that would take far too much time, I will try and cover some of the main points that I made in my paper. Starting with the general shape and structure of proteins and how they gain this shape and structure, and moving on to the function of DNA ligase, then to the structure of DNA ligase itself, and I will end it with why I believe DNA ligase is an important protein for all organisms. So to begin, what is a protein? A protein is formed when a series of amino acids come together in a very unique sequence and bind to one another to form what is known as a polypeptide chain. This polypeptide chain will be bent and shaped and altered into a variety of shapes known as either alpha helixes or beta pleated sheets. The total number of the beta pleated sheets and the alpha helixes, as well as any other little unofficially recognized folds and loops in the chain, are referred to as the polypeptide's 3D conformation. At this point, it could be considered a protein, but sometimes an extra step is taken, and multiple polypeptide chains that have reached their full 3D conformation will come together and form a more complex protein. Proteins will demonstrate time and time again that their structure, which is very unique from one protein to another, will directly impact how they function in their given tasks. So to move on, I would like to talk first about the function of DNA ligase, and then explain how this function is carried out through its structure and what its structure is. DNA ligase is a protein responsible for binding together fragments of DNA. It is most often seen in genetic replication or gene replication, where the DNA polymerase II will read the lagging strand and produce these little Okazaki fragments instead of a continuous strand. At that point, it's the DNA ligase's turn to come in and take these disenfranchised strands and bind them together into a much more useful continuous 5-3 strand. In addition to being a key component of gene replication, it is also a driving component behind DNA repair. DNA ligase is primarily responsible for the repair processes in both DNA single and double strand breaks, as well as being responsible for nucleotide and base excision repair. So pretty much whenever DNA is broken, DNA ligase is going to come in and fix it all up, no ifs, ands, or buts about it, and no unfortunate plumber bills at the end. So to reinforce this idea, instead of just telling you that it's responsible for DNA repairs, I'd like to point out a study done at the Graduate School of Nanobiosciences in Yokohama, Japan, when they were specifically looking at double strand breaks in the DNA and how they are repaired. The researchers noted that there are two main ways in which the double strand break will be repaired. The first of which is homologous recombination, and the second is non-homologous end joining. More often than not, non-homologous end joining is what occurs to fix a double strand break. And with non-homologous end joining, you'll find that DNA ligase is a key component to the entire process. So what the researchers did is they took an isolated human genome and they used a series of genetic knockout techniques to remove the sequence that specifically coded for human DNA ligase 4. When they removed human DNA ligase 4, they saw that there is a complete stop in all non-homologous end joining, meaning that DNA ligase 4 in humans is not just an important factor, but it is the driving factor between non-homologous recombination. And without it, non-homologous end joining, because it occurs so much more frequently than homologous recombination, would mean that more often than not, double strand breaks would go unrepaired in the DNA. And I'm certain I don't have to tell you how bad it is to have damaged or broken DNA. Broken DNA cannot be properly expressed, it cannot be properly replicated, and quite frankly, I doubt any organism would survive if it could not consistently fix these breaks that occur on a large scale. Now, the DNA ligase gains its ability to repair this DNA because of its shape. DNA ligase is found in all organisms, and many organisms have multiple forms of DNA ligase, so its exact structure is going to vary quite significantly depending on which specific form you're looking at, but they all have the same general shape, in that they are all enzymes made up of 3 subunits that come together in a ring shape. 2 of the subunits will remain consistent no matter what form of ligase you're looking at. Those 2 consistent subunits are NTase and the OB fold. They will sit over top of a DNA strand, and when activated, the 3rd subunit, which varies from creature to creature, in humans I believe it is referred to as the DVD complex. Again, the DVD complex will not be consistent. Its general shape and the ratio and number of helixes and pleated sheets will alter depending on whether or not you're talking about human DNA ligase 1, 2, 3, or 4. But the DVD complex, or just in general that 3rd subunit, will come together around the protein, and it'll form a ring with a hole in the center with which the DNA will sit in. Once that ring is formed, the high energy cofactor ATP will bind to the active site of the protein, and it will initiate the binding process. And slowly the protein will move across the DNA strand and phosphorylate the 5' end to the 3' end to form a continuous strand. So essentially, because of this shape, it allows it to surround the strand completely and bind it all around, not just one portion over the top or to the side. And thanks to this ability, DNA ligase is solely responsible for maintaining genetic integrity within all organisms. Without it being able to go in and repair all the damages done, quite frankly, I don't think life would have been able to reach the point that it has. I don't think we would have gotten much farther than a few single-celled organisms if we all were just meandering about with slowly corroding DNA with no way to really fix it. So yes, to summarize, proteins are made up of a variety of amino acids that form a polypeptide chain. This chain gets folded and shaped into a very unique structure, and this structure will go about to aid that protein in performing its job. In the case of DNA ligase, its job is to bind together fragments of DNA and keep them as a continuous strand. This is implemented in gene replication and general gene repair, well, DNA break repairs. It is able to repair these DNAs and bind these fragments together because it is a ring-like shape that sits around the DNA and allows it to interact with the entire structure of the DNA strand, not a simple portion of it. Otherwise, we would have a very sloppy repair job, something I would do, and we would have simply, if it sat over the top, one side binded to the fragment, which is not a very structurally secure way to fix something. So yes, the DNA ligase forms a nice little ring that completely envelops the DNA strand and allows it to bind it to the other fragment. And because of its ability to fix these broken strands, it allows organisms to maintain their genetic integrity. When the DNA is good and stable, you can express that properly, you can express the DNA appropriately, you can replicate it appropriately, and you can allow life to continue. That is my presentation. Again, this has been Megan Lahan. I was assigned, once more, DNA ligase. Thank you for watching.